Okay, so today we are going to do the MTT assay experiment to study the chemotoxicity of the doxorubicin on the HeLa cells. Okay, so we already saw the HeLa cells last Friday, and we cultured for about 3 days. And now I'm going to take it out, and then we are going to follow the lab handbook of the MTT assay for this experiment. Okay, so well, I'm going to take out the cell. Okay, we can have a brief look under the microscope. Okay, not so many cells. Okay, maybe we now have a confluency of about maybe 40% maybe, okay, by estimation. Okay, but it should be sufficient for our experiment. Okay. So next, we wanted to get rid of the old medium and add PBS to wash 2 times, and then we are going to add Trypsin EDTA to detach the cells from the dish bottom. Okay, because the HeLa cells are tightly attached to the dish bottom. Okay, you may remember why we wanted to wash the cells before we add detaching agent. Okay, this is because the FBS, will reduce the function, the activity of the detaching agent. Okay, that's why we wanted to remove the medium and any fetal bovine serum from the cultured medium before we add, and also wash with the PBS before we add the detaching agent. Okay. Okay, we are going to add PBS to wash 2 times. This is our sterile PBS. Since the HeLa cell is tightly attached, so you can do this kind of addition of the PBS. Not like a HEC293 cells, okay? And we are going to get rid of it. This is to remove fetal bovine serum, okay? Okay, we are going to add 1 more time. Okay, next we are going to add a Trypsin EDTA, which is already warmed. Okay, remember, before we move anything into the cell culture food, we want to spray the alcohol and wipe the outside surface. And always open the cup inside the cell culture food, okay? And we only need to add 2 milliliters to cover the whole dish bottom. Okay, we are going to put it into the incubator and keep it for maybe a few minutes. So enzyme trypsin EDTA works the best at 37 degrees Celsius. Okay, so after a few minutes of the incubation, I can see most of the cells already detached. Okay, this a little cloudy thing from here, a little bit of cloudy. That is white colored. That is the cell. So what I'm going to do, I wanted to add a fresh medium that contain fetal bovine serum to stop further action of the trypsin EDTA. Okay. And this complete medium doesn't have color. This is because we wanted to use phenol red free medium for this MTT assay. Okay. Okay, I'm going to add 10 milliliter of the complete medium that contain fetal bovine serum to stop further action of the detaching agent. Okay, and then I'm going to collect it. Okay, into a 15 ml conical tubes. Okay, you can spray the bottom of the cell culture dish, okay, to collect all the cells. Okay, and then we are going to centrifuge to pellet the cell. Okay, 1000 RPM, 5 minutes. Okay, so after pelleting, we get a small, pellet cell heads here. Okay, so now we want to get rid of the medium, okay, and then we are going to resuspend it in the complete medium, and we wanted to count how many cells we have. Okay, so now we wanted to add complete medium. Let's add 2 milliliters, okay, phenol red free medium, okay. Okay, so next, I'm going to use the 1 milliliter pipette to get a single cell suspension, which means I will do up and down pipetting to get the disrupt the cell pellets to have the single cell suspension, okay? And after that, I'm going to further dilute the cell with the complete medium for cell counting, okay? So I'm going to take out an epidoff tube. Okay, you better not to put your, hands to grab the epidoff tube. You can do this way. We just need one. Okay. Okay, in order to accurately count the cell, we need to have a good cell suspension. Okay, and the next I'm going to add 120 microliter of the complete medium. And 20 microliter of the cell suspension in a mix. Okay, and then we are going to add 200 microliter of Trypan Blue. Okay, so Trypan Blue will stain the data cells. For cell counting, we can do it outside of the cell culture folder. Okay, we can do it here. Okay, this is a 0.4% Trypan Blue. Okay, and we are going to mix with the cell at a 1-to-1 volume ratio. Okay, so which means here I have 200 microliter of the cell. I'm going to add 200 microliter Trypan Blue. Okay, and then mix a little bit. Okay. Okay, and the Trypan Blue will be able to permeate into the data cell membrane. Okay, if the cell membrane is intact, okay, it won't penetrate into. Okay, this will allow us to exclude the data cells. Okay, so next we are going to use the Hemocytometer. Okay, this is the most traditional way to count the cells. Nowadays, there are also some automated cell counter. Okay, we are going to use the traditional way to count the cell. And this is the Hemocytometer. It has two sides. Okay, and we need to use this cover glass after we put the cell. Okay, you can see here there are some cuvettes. Okay, V-shaped cuvettes to allow us to load the cell. We already clean with 75% ethanol, you know. Okay, so we are going to assemble the cover glass with the Hemocytometer. Okay, and we are going to load 20 microliter, okay, or 15 microliter. Okay, let's do 15. 15 microliter of the cell. Okay, before you add it, make sure you have a good cell mixture. Okay, and then you can add from here. Okay, so it will spread to the entire space. Okay, and you can check under the microscope. Like I said, we don't have too many cells. Okay, I only saw 3 cells in this 16, in this 4x4 grids. Okay, so basically under the microscope, okay, in order to count the cell, we are looking for the 4, okay, at the 4 corners we have 4 by 4 small grids. Okay, and you want to count how many live and dead cells in each of the grids. Okay, here I counted. In the right lower one, I got 3 live cells, 0 dead cells. In the right upper one, I get 6 live cells, 0 dead cells. In the left upper one, I get 5 live cells, 0 dead cells. In the left lower one, I get 9 live cells and 1 dead cell. Okay. Now we wanted to calculate how many cells we get, Okay, so how to count? We wanted to add the numbers up. Okay. Non-standard cell. Okay, so non-standard cell, which means live cell. Okay, let's have an addition. Okay, 5, 6, 11 plus 9, 20 plus 3, 23. So we have 23 non-standard cell. Non-standard plus blue stand. Okay, we have 1 blue stand, and we times 100%, 24, 23. This will be the live cell percentage. Okay, we can calculate later using the calculator. Okay, and live cell density, we need to divide 23 by 4 times 10 to the 4th order times 20. Okay, equals 1.15 times 10 to the 4th order, 10 to the 6th order. Which means in that 2 milliliter of the cell suspension, we have 1.15 million cells. Okay, so the unit will be cells per milliliter. And now we wanted to adjust the cells to 200,000 cells per milliliter, which means we wanted to dilute. Okay, let's calculate the dilution factor. This number divided by 11.5, 10 to the 5th order divided by 2 times 10 to the 5th order, we get 5.7 or 8, 5.8 times. Okay, so which means we need to dilute the current cell in order to adjust the cell to this density. Okay, so how to adjust, how to do the dilution? We already have 2 milliliter inside. Okay, which means we just need to add 2 milliliter times 5.8 minus 1. Okay, which means 4.8, 9.6 milliliter of the complete medium. Okay, and the cell concentration will be 200,000 cells per milliliter. Okay, and then next we just need to see the 100 microliter of the cells per well into the 96 well plates, and then add the different serial dilution of the doxorubicin into each of the cells. Okay. We need a 9.6. Add into LSA. Okay, and then we are going to pipette up and down to have a good single cell suspension. Okay, we are going to add 100 microliter of the cell. Since we don't have too many wells to add, I'm going to add one by one. But we can use multi-channel pipette to add. Okay, cells is very easy to sink. So before I add, I'm going to pipette one more time. So we want a good cell suspension. going to add from A to C rows and 1 to 6 columns. Okay. Okay, and the next I'm going to add doxorubicin drugs, okay, which they are already prepared at different concentrations. Okay, let's add from high to low concentrations. Okay, so what we are going to do, we are adding 11.2 microliter. Okay. Okay. Okay, I'm going to first add the highest one. It has some color because the doxorubicin at high concentration has some a little like red, brown color. Okay. I'm going to add into the cell culture solution. Okay, and also pipette a few times. Okay, I'm going to change the tip. Okay, so now we are putting the cover and slightly tap a little bit. And we are going to put it into the incubator in a culture for 2 days. And then use the MTT assay, MTT kit to measure, the relative live cell numbers. Okay.